head plant virology department and chief virologist, German collection of microorganism and cell culture, Germany. Prior to that, he worked in the world as a World Bank advisor to the National Plant Quarantine Services in Nigeria, and then subsequently, prior to that, as a plant virologist, Agriculture Canada Research Station, Canada. And his area of expertise in viral diagnostics, virus diseases of tropical roots, tuber crops, banana, virus vector interactions, virus resistance, and biotechnology. Without biotechnology, virus cannot be worked out very nicely. And professional achievements, a lot of professional achievements are in his credit. He handled several research projects, including the projects financed by the big organizations like Bill Gates and Melinda Foundations, EU projects, and he served as the editor-in-chief, Journal of Plant Disease and Protection, and associate editor, European Journal of Plant Pathology. He is the member of American Phytopathological Society as well as the German Phytomedicine Society and General Microbiological Society, that is Society of General Microbiology in Germany. It is very famous society, and published more than 50 research papers, several books and technical bulletins, mentor of more than a dozen students, PhD students, and supervisor of several international PhD students. That's shows his vast experience internationally, and visited several countries. And today's topic of his lecture is Cassava brown stick management and control options in the morning. Disease sir has pointed out this is another emerging disease in the cassava, which threatening the cassava industry. So now I invite Dr. Stephan. Dr. Stephan, please, now floor is yours. Stephan, please unmute yourself. Unmute. Dr. Stephan, you muted. Dr. Stephan, you are muted. Please unmute first.
So, can you hear me now?
Now it is okay. The slide is also visible. Oh.
So I started again, please, and apologize for this.
Okay, thank you. Now.
Yeah. Good morning and good afternoon, everyone. Thank you very much for the consideration of my person. And I would like to talk today about my heart disease and these cassava viruses. I had changed more a little bit my topic because my friends and colleagues already spoke a lot about cassava viruses. And so I am now taking with you on a ride, and I'm not quite sure where it will be. So I would like to first show you and entertain you with some pictures. And the reason why I do this is because I would like to contrast these two diseases that are on the African continent. And maybe we can then, at the end of my talk or during my talk, realize why it may be specific to Africa. So we are dealing with disease that everyone has spoken about. We have made a lot of experience with this because of a mosaic disease and a recently emerging disease caused by two viruses, which is because of a brown streak disease, and that causes really this quite serious destruction to the roots. So what I'm talking about today is something that's a bit far from my home front, with these diagnostics and quarantine and all kinds of facts. So I talk about the diseases, two diseases.
Please,
and I talk about.
Please, Dr. Stephan, please share your slide. Your slide is not visible.
Why is the slide not visible?
You know, it is not visible. You have to share it. Open and share. It has not been shared.
Somehow I need to. Why am I not? Oh, I thought I was there.
Yeah, now it is. Now it is okay. You have.
Oh, sorry. That was a technical.
No problem. Yeah.
That was a technical.
You can put in the slide. Yeah, that's good.
So these two diseases, one is causing a serious reduction in the root size, and the other one has this destruction route, while the plant otherwise is going quite well. So today I would like to talk something that's a little bit distant from me, because I would like to talk about management. And the reason why I'm doing this is because often in the discussion that we have in Europe, when it comes to quarantine, when it comes to new introductions, we spend a lot of time into risk assessment before we come to the management aspect. So I would like to talk about the biology of the viruses. So not diagnostics, but not about the diversity of viruses. I would like to talk about the biology of the viruses, go back to something that's called entry, establishment, and spread, because this is something that we have to evaluate when it comes to management of a disease. After that, I would have to look at the control options, and these control options can be, of course, different to the two diseases, and so maybe all of us can then go and think about what we can do from phytosanitation to virus resistance. So as I said, I've changed my topic a little simply because my speakers have basically taken talking, and the first thing I would like to make is this is not a pandemic. So it's an endemic disease in Africa. So, and we know it quite well. They have made their nice extension of their distribution, which was based on the fact that the level of resistance at the time in the 80s in Africa was pretty low. And certainly we have a Bemisia tabaki that can readily spread the disease, at least in Africa. So the only thing what we cannot differentiate well, this is to differentiate between primary and secondary spread. But I know from my own experiments and experience that in Africa, Bemisia tabaki is super competent in transmitting the disease. But because the disease is open, in the open field, we really have limited control options. We cannot do like we do in in the southern part of Europe with a similar disease, a tomato yellow leaf curl disease, that we just pack in large greenhouses to have the whitefly exempt. So all what we have is we're looking for virus resistance. And just to make this comment, virus resistance is found by looking for it. So if you don't have it, you can do as much GWAS and GPS and whatever you like to do, you will not reach anywhere. So this was basically the game changer for the cassava mosaic disease in Africa, that CMD2 and CMD3 were found, because before we had this recovery resistant for which every breeder in the past has elected for, but with the CMD2, CMD3 resistance, or CMD2 was found, CMD3 resistance was spread, actually the situation became solved more or less. And if you look at these situations here, you will see a variety called TME419 or Obama in Congo since 10 years is essentially symptom and virus free. And also you have enormously good varieties that are more or less withstanding almost any harm that could be done with new viruses or new virus variants. And the interesting thing about this, and I'd like to contribute to the discussion for this more, this is a dominant resistance. So this dominant resistance, which in plant biology is a little bit different, G99. So this dominant resistance essentially does not allow the virus to replicate. There's things missing that the virus actually needs after might get an initial virus replication, but this replication stops. This initial virus replication probably is because of the heterozygoty of the cassava that is in concern. Now, we have the cassava brown streak disease, and this is really an outbreak. So here we have an outbreak in Eastern, Central, and Southern Africa. James Lake, Lava have sort of shown this very nicely, how this disease spread, but we have to ask ourselves, the question is, what is really the biology of this outbreak? Yeah, so how is this outbreak characterized? Certainly we have two distinct virus species. They cause similar symptoms at the end of the game. They have a very similar result on the roots. They destroy really the roots with heavy necrosis symptoms in an otherwise very well developing crop. So the crop at the end of the year, at the end of the season, looks very super. Most of the crop has already dropped the leaf, so you don't see symptoms. Yeah, and you also can get quite interestingly big tubers. However, if you open the tubers, you will get this type of symptom that James and others have already shown. The interesting thing is to look at the role of the Bemisia as the vice vector, because if we make risk assessments, okay, we always have to say what is the biology and what can we prevent and what can we not. So for CMD, which grows in the open, cassava grows in the open field, it is very difficult to have measures like roving in order to prevent the spreading of the disease. What we also have, and I'm sorry, Mahodi, that I'm going this way, there is no resistance against the virus causing DBSD in Africa, despite the fact that there is some quite good and vigorously growing lines, and I would like to go on the risk of those lines for the cassava production in Africa during my talk. First of all, I would like to look at Bemisia tabaci for virus spread. And to warn you, this is like a work in progress and as we fly. So as you know, I mean, the Ebola to which the two viruses belong to are transmitted semi-persistently. They have very short return times, and thus they can have a limited dispersal only. The interesting thing is when we study a cassava brown streak virus and we contrast them with a very similar virus, which is called cucumber vein yellowing virus, which is very readily spread. And when we looked at those actually the code proteins, we realized that in a part of this code protein, we can abolish the ready transmission of cucumber vein yellowing virus, and this part then resembles cassava brown streak virus. So I'm very clear with the fact the virus is transmitted by Bemisia tabaci, but very, very, very inefficient. And however, it doesn't matter because we have seasonal upsurge of population as shown in one of the slides of James, particularly after the long and short rains. So all in all, we can say that this particular disease has shaped, or this virus has shaped towards a vegetative propagation. And that's also clear because the virus might not need it for its survival. So as we've seen in other viruses, they just drop a factor that they may not necessarily need to sustain in the environment. So this can provide us the first argument for intervention. So these viruses may not necessarily be as easily Bemisia tabaci transmitted as often it is spoken when we talk about CMD. The other fact that we have basically studied in the past several months, I would say, we have looked at quite a large number of genomes that we have produced. And we made an analysis of their spatial and temporal distribution. And I'm sparing with you here a moving map, but what you can essentially see is that the viruses were reported around 1906 along the Tanzania-Kenya coast, and from there made a spread. You see that from these centers. Yeah? So you see spreading back and forth also from the centers. And these centers are areas where breeding activity has been or is done. And we see that the recent movement into the western part is most likely man-made spread. And of course, human-assisted spread is one of the main keys when you talk about cassava and cassava virus movement. However, if you talk about human-assisted spread, this is something that you can also, let me say, intervene with. So from those sequences that we have, we can more or less follow each of those isolates, I would say, specific isolates, even traveling from a particular area to reach another one with exchange of germplasm. So when I summarize now my argumentation for entry, we have to say that it is really a fact that the planting materials plays a major role, and this might not surprise you, but I just say it again that it keeps us in our mind. Our question is just, are there other virus-infected sources? Because a virus that is not necessarily circular transmitted must have some sort of an alternative host. And we look at establishment. So entry, we could prevent. So making border crossings impossible, which of course is in Africa something that is out of any reach. So entry is with infected plant materials, but we also have to say, can it really enter also with something else? And how does it establish? Does it need cassava throughout? Can other plants in the environment function as virus reservoir? What you see here actually is the plant de la Roussis in Congo. And this a nice plant that you see here is Partenio. This was recently introduced. And people who work in India, not on cassava, know what this Partenio means, because it was basically the host for toscoviruses and tobacco streak virus, causing quite severe outbreaks in cotton and brinjal. So if we look at spread, long distance spread is by vegetative propagation materials, it's clear, but the spread is from focal points. So it's not like range expansion that might come from a whitefly. And in fact, a whitefly can fly very fast and very far, because it can also fly, being a viruliferous, it can fly with a track over long distances to meet into new areas. Okay, so we are left with the role of the insect vector, and James Lake rightly put it, it is an enormous amount of insect, of Bermesia in recent times that we see on cover as it was never before. So what do we leave with control options? So what do we have in control options that we can tackle for brown streak disease that we might not have for the mosaic disease? First of all, phytosanitation. And here I would like to bring you to sort of another place that's close to my heart, and this is an island on the Lac Kivu, on the Kivu Lake. And this island, there was, we had a big activity to bring in resistant, CMD-resistant varieties, and we followed this for quite a long time. And only in 2017, with improved material, inadvertently, cassava brown streak was introduced. In a community action, which is probably very, very difficult to do, but we succeeded in convincing the farmers that what you see here on this picture is to do eradication. And it was a very painful process, because eradication and roguing needs that you have a seed system. You need to replace what you have. You need to replace something with something better, or to replace something with something healthy. And that was really on this island a very difficult approach. But up until now, and I was just recently there in 2021, this island is essentially virus-free. And this gives us a place for the production of healthy cassava material in a disease-free zone. So for any mitigation, we need seed systems. And of course, we can think about cropping cycles, but these cropping cycles are always a little bit problematic, because simply in Africa, you have 360 days of the year, there's cassava somewhere in field. What I personally think is the worst is tolerant varieties. Because what we see that tolerant varieties, these Narocas 1, okay, they have a very vigorous growth here, grown in large scale, on a very large scale, to produce varieties, improved varieties for farmers, to give them to the farmers for planting. But what you do here with this particular crop, you basically spread 100% of the infection. And surveillance for me is a very easy thing, because in half a year you will see that the zone has moved a further 100 kilometers to the west. So if you have anything better, try anything better. But is there anything better? I don't know. What about virus resistance? Well, virus resistance only is not coming with GWAS, it's coming with the sources of resistance. It's coming with also very competent screening protocols, because Cassava brown streak virus is about the most difficult virus disease that I know. Cassava brown streak viruses are one of the few viruses, and I only know 2 others that go on the roots. But they can also stay on the roots, and we never see them, even by qPCR. Okay, so we need to have screening protocols that don't take 5 years in order to find something out. So, and we are dealing with an outbreak. We have to have a short breeding cycle. We must bring something to the field. We cannot wait until we have sort of researched and found out the last specific for Cassava brown streak viruses and the cassava and so on and so forth. And this brings me to now the change of my gear. This brings me to the introduction of Dr. Samar Sheet, who has worked in my department to work on Cassava brown streak virus resistant cassava. And actually what I'm talking about now, search for virus resistance, identify virus resistance, evaluate the virus resistance, and to investigate the resistance mechanism is not my piece, but it's mostly the work that Samar and the team has done. And despite the fact that we have poor sequencing, you can do that quite easy if you want. Okay, so you take out a grafting knife, you develop a very, very efficient infection method, here using 2 buds of an infected plant. You know that what is the most severe virus for Cassava brown streak virus, and when it develops symptoms, you can just say, this might probably not a virus resistant plant, or at least the virus resistance is not correlated with symptoms. So we can do grafting, and this is exactly what we did, because despite the fact that we have very competent equipment around and infectious virus clones and so on and so forth, this is not efficient enough if you go for large scale. So the first what we did is we looked at cassava viruses on hand. It were about 50, 60 different varieties we checked, and what we found is that most of them wilted and died. Many of them caused symptoms on leaves. Here you see these necrosis on the midvein from the moving virus, and some showed mild symptoms only, and these were the ones that we can call, or that were called tolerant varieties. Also a very, very dangerous name given and used, which I have to say I'm not very pleased. So very clear, all African lines and breeding lines can be infected with Cassava brown streak virus, and this extends also to Ugandan Cassava brown streak virus with some exemptions. So one of the reasons why then we were all stuck was simply there is no resistance. And of course, this gives a lot of way to just say, okay, let's do some biotech and make GMO cassava and so on. But this was not the work that we were up to, because we were asked then to look at resistance in South American cassava germplasm. And this is actually the biggest wild card I've ever been, and the biggest adventure I've ever been into, because we are dealing with an African virus, and we are looking for research for resistance in a South American germplasm. And the South American germplasm is huge. It is held and kept at CIAT, and it has a core collection of 600-700 cassava lines from all over South America, from Peru to Ecuador. And using our very competent
let me say, infection method, okay, we have found materials that didn't show symptoms, and by qPCR that also we never found virus. We had symptoms on roots for others, but never on leaves and never on the upper plant part. And of course, most of them, they just died. Most of them, they just succumbed to the disease. But the few that had no symptoms and we saw that they were immune to virus, we realized, wow, this can be an interesting aspect further developing virus resistance. And certainly we knew that most of the people will not believe us because what we do in the laboratory is not something that you find in the field. So again, we took another strategy, and the good thing, if you are a dinosaur in plant research, you still know how to do some grafting. And as you can see here, okay, you take what you call a resistant plant, you put a susceptible plant as a side graft, you infect the resistant plant. If the side graft gets an infection, you know that you have introduced a disease. If the resistant plant stays resistant, okay, and of course you can just say there must be something. So you really grill those plants, and you can do this vice versa, and you can even do and follow virus movement as we did. So this was the setup of the cassava resistance research that we were doing. And of course, Samashid again came up and said, okay, we have to find a little bit more interesting and advanced techniques, even techniques that will probably or can probably beat qPCR and LAMP and all kinds of type of assays. And because we were interested to see the fate of a cassava brown stick virus in a resistant plant, that was negative for qPCR. So we have developed a method that you can see here. This is a leaf of a cassava, this is a cassava leaf, and these are the mid veins and the side veins and the minor veins and so on. And you see how these red dots, each reflecting one single virus molecule, ooze out of these mid veins and minor veins into the parenchyma to replicate in a susceptible plant. So this is what you get when you do something that's called RNA scope. In situ hybridization, you can follow it. And when you apply this to a cassava plant, and this is what people call a susceptible cassava plant, you see that in these leaf tissues, that the leaf tissue is full, is exactly full of those little red dots that represent virus molecules, and they form, of course, clusters when we have much more virus replications. There is. So on a susceptible plant, and now cassava is susceptible to us, you will see lots and lots of red. So again, in a leaf section, as you see here, so this is our tester line, you will see an Albert, highly susceptible to CBSD. Essentially every single leaf cell, every single tissue is full of this virus. And even in a plant that is called resistance, like Namikonga, where you have areas that are not invaded by the virus, in areas where there is invaded, there is heavy virus replication. And this is what I have to show you instead. So we have this South American line sitting on a virus source. So we feed this virus constantly in this line. We can trace it into some areas of the leaves, the mid vein, but there is absolutely no virus replication. And this is supported by qPCR, of course, where we can now, this is mid vein, yeah, where we can do some qPCR. It's very erratic, but we can do some qPCR in which we find traces of virus, but these traces are only there because we put this in, because this is still sitting on a virus-infected source plant. Now, to summarize, in a virus-susceptible plant, the virus is all cell types. It replicates in the parenchyma cells, it replicates all over the place. While in the immune line, there is no replication, but the virus can only move in the external phloem. To remind you, cassava has two phloem types, which I show you here. This is the internal phloem, and this is the external phloem. And this here is the cortex. So we have two types of resistance, an immune line, and we have a virus resistance that basically is restricting the virus to the root. Of course, for us, this is not very funny because we want to eat the root, but for the cassava plant, this is a resistance mechanism that is quite interesting to follow. So the cassava responds, this cassava responds to the virus infection by restricting the virus to the root tissue, also indicating that between the roots and the leaves, we have a very, very significant distinction, and that probably, and we have much more indications now for other viruses and other diseases, that the root system plays a major role in the resistance of a plant against pathogens. And of course, if we look at the root of the immune line, this is a susceptible one, and this is a root restricted one, and this is the immune line. In the immune line, you barely find it. You find it only because the resistance source is still on. And in the phloem-restricted root line, you will find virus replication by indication of these red dots. Of course, we have extended this. So we have now said, okay, let's see whether we have broad-spectrum resistance. And here, these are all roots that we cut open that were infected with different viruses and different isolates belonging to Cassava brown streak virus group. Now, to summarize this part of the work is we have found at least 3 lines that have a broad spectrum resistance against Cassava brown streak virus. And these, I mean all. These 3 lines, I have to say, there's probably much more lines because we have brought some of those lines into an African situation, and in an African situation also our colleagues send us some of those back. Some of those lines that are root restricted are not, we are not capable to infect, which also shows that for virologists that there is a difference between infectibility and resistance, because some plants cannot very easily infect it. And this is what I just got from my friend and colleague from Tari, Tanzania. Some of those lines here, this is the one that we have here, these 2. And you see that they're also making quite appreciable root sizes compared to a South American tester line, Group 40, that we have used to also as an infection control. So I have to say that there is virus resistance against Cassava brown streak virus, and this virus resistance is also holding against a variety of those. And certainly there's also differential resistance. So differential resistance is also a case in the South American lines, but also some of the African lines have a differential resistance that one can basically also draw on. So we have a broad spectrum immunity, and I speak the word immunity because we know that the viruses cannot replicate. So South American varieties might have a good chance to provide the resistance results for Cassava brown streak virus in Africa. However, what is their problem? The problem is that if you bring South American Cassava varieties into Africa, most of them look like this. They just get toasted of CMD. So if they even get close to the African CMD, and this is East African Cassava mosaic virus that are really, really severe. Most of them, as you can see, get really destroyed by this. And here we had to also change the gear. As I said, I'm not very patient because I'm old and we are dealing with an outbreak. And we asked our friends from SIAD to do some crosses, of our best mothers or best fathers or best resistant lines here, with crosses that have a CMD2 resistance. And we got those seeds and we tested those seeds in our farm in Germany. And this time we changed our strategy. And as you can see here on the timeline, Okay, on this timeline, we were essentially capable of testing the material in a very short time. It took us 2 months to get into the seeds to the little plantlets. It took us then our efficient grafting line. Most of the plants die. The ones that do not show symptoms will be subjected to EACMB infection, and the confirmation cycle starts. However, we almost instantly introduced open field. And I think this was the key to success. And of course, this was done with our colleagues, for which we have very, very long and standing experience, so that we can almost instantly bring this at a very early stage into the field in order to see whether our hypothesis is right. So we now can say that we have CMD and CBSD resistant cassava. This is resistant cassava, and this does not mean that this is, has other very good agronomic traits. We started out with seeds because, you know, and Dr. Sheila spoke about this, cassava doesn't flower very well. Some of those lines that we brought into Africa die of CMD, where they don't make seeds, and others just are infertile. So we had to go back to our colleagues to SIAD, get some first crosses in order also to prove whether we are right. We tested them for 2 resistances, CMD and CBSD. And we found out that also cassava brown streak virus resistance is a dominant trait. So it's a dominant trait, just like CMD. And I think this probably is a gain of function situation here, because as we see with other dominant traits, there is probably an in-progression of additional material that provides this resistance. So we learned how to do fairly quickly to screen, because this is very simple and very important, because field screening for CBSD takes many seasons because of the very different infection biology. We deliver this resistance into the field and we propagate this without having SAR technology available. We also showed the guys and our friends of how to test for it in the sentinel sites. And we are looking forward to see our results, for which here on this side, we already have the first quite good indication of a healthy plant. We have advanced the screening to improve, first of all, the precision, because the precision means we know what type of virus we are dealing with, and also the pace, by reducing this 2.5-year cycle into a cycle that already goes to the confirmation after the 2nd year. And I think we think that in the future, this can, of course, provide some major advancements into cassava brown streak virus and CMD virus resistance. So let me summarize. First of all, cassava brown streak virus have a super complex infection biology. It's really, I would say, very easy underscored. It takes a very long time, even for a variety like KBH 2618, to even show symptoms. It can show up symptoms 1 year after you have infected it. And before this 1 year, you don't see, even by qPCR, that you have a virus infection. So the spread in the field, the human-assisted spread, is very difficult to control, because even if you do diagnostics, you might have a big chance to oversee. There is control options, and these control options probably are also underscored. So there is possibilities to do phytosanitary, as we have seen, but the feasibility under African situations is very low, because there's constantly people moving in and out in order to bring new virus-infected material, in particular because cassava brown streak infected varieties can look very good. They can look very vigorous, they have very strong stems, they can do very well. Of course, we need seed systems, but we also need these seed systems in pest-free zones in order to produce enough material to replace infected materials. And I don't think by myself that can be done centralized, as I rather think that we really have to look at decentralized opportunities, so that we can develop farms that we support with certification system in order that the material is quickly replaced. There is virus-resistant cassava identified, but definitely under African conditions, if you don't have CMD, CVSD resistance doesn't mean anything. And of course, I have overgone this, yeah? But certainly you have to ask yourself whether CVSD is really, does have to be a pandemic situation. It's simply because we are not sure whether in the West African conditions are suitable for CVSD viruses to be really proliferating and to be living happily. So we have developed new approaches to resistance screening processes, and we have already showed in 2019, before the pandemic started really for the COVID, that this can be done in the field by doing field type crafting. And it's just a matter of commitment that you can get this done. However, if I look at what we have done in our class houses, it requires really competent hands and will, and you can do the first type of screening for virus resistance already under laboratory conditions. What I also have learned is that we have to immediately integrate stakeholders, and because this is very simple, because the communication of what we are doing is often, even as a scientist, is a very difficult one and a very lengthy process, because I really have to say that despite the fact that we just sometimes speak a different language. With that, I have to come to thank the people that provide us the money, and in particular, the 6 girls that are working with me downstairs. They can do the NGS sequencing, to the tissue culture, to the crafting, to the RNA scope. So I'm basically can lean back and just hang around and see what other people do. And all of the friends that I put here that help us to deliver. So in this short version of what I wanted to tell, I thank you very much for your kind attention. And if you really want to hear more a little bit about that, I can invite you to a talk that I give next week, where you can see even my farm. So many people who might think that I'm lying, I will be able to show you my farm, having a workshop next week where we only speak about cassava brown streak virus and cassava mosaic in Africa. Thank you so much.
Thank you, Dr. Stephan.